Hi everyone, this is Lauren, and this is my group Advanced Concepts presentation number 8. This is just a presentation of my poster board for the labs that we've been doing the past couple weeks. You can see the whole poster here. It's really big, so it's hard to kind of see everything, but I'm going to zoom in on a few things and talk about exactly how ATP and some of the other nucleotides are formed through these pathways. So just moving in here, we're going to start from glucose. And as we talked about in the beginning, we can form ATP by going through cellular respiration. So you see proceeding through the glycolytic pathway, we form pyruvate, which enters the pyruvate dehydrogenase complex, forming acetyl-CoA. The acetyl-CoA enters the citric acid cycle, which then enters the electron transport chain and the ATP synthase enzyme, which converts the protons to approximately 30 ATP through this process. And one of the intermediates along the glycolytic pathway is glucose 6-phosphate. So depending on the needs of the cell, we could actually proceed through a few different pathways. So we can of course go through the glycolytic pathway, but also this glucose 6-phosphate can proceed along the pentose phosphate pathway, which is really important for synthesizing nucleotides, which is important for making DNA and RNA. So that's important especially for cells that are rapidly dividing. And so we see the glucose 6-phosphate, which can enter the pentose phosphate pathway that we have here. There are two phases to the pentose phosphate pathway. There's the oxidative phase, which forms NADPH, which is important, an important reducing agent for a lot of biosynthetic processes. And there's the the non-oxidative phase, which actually involves the interconversion of sugars with different carbons. So you can kind of see that interconversion here, and that is very important for forming the base of nucleotides. So the main thing formed in the pentose phosphate pathway is ribulose 5-phosphate, which can isomerize to ribose 5-phosphate, and that is the sugar that's used for RNA and DNA synthesis. And like I said before, depending on the needs of the cell, then we may, with this glucose 6-phosphate, proceed along the glycolytic pathway, but also for rapidly dividing cells where it's important to create these nucleotides. There are some intermediates along the glycolytic pathway that actually can enter the pentose phosphate pathway and form more ribose 5-phosphate if needed. Up here you see that ribose 5-phosphate can be converted to PRPP, or phosphoribosol pyrophosphate, through the ribose phosphate diphosphokinase enzyme, and that's really important for forming the bases of nucleotides. So from there, we're going to go all the way over here. And these two just basically talk about how we use that PRPP, and also when we talked about how we start from glucose and CO2. The CO2 is important. If you look up here, CO2 is important for the formation of the pyrimidine bases, because that you can see all the steps that it goes through to actually convert to uridylate, or UMP. And then UMP here can convert to the pyrimidine bases. So these are the nucleotides that are based off of pyrimidine bases, which is UTP, CTP, and TTP. And then also we can see from PRPP the steps that it goes through to form inositol, which is important for the formation of the purine bases here, like ATP and GTP. So these nucleotides here, these 5, can actually be formed through the de novo synthesis of the purine or pyrimidine bases. It can actually be formed too through salvage pathways, where there's a base that can actually be attached to a ribose sugar, and that's really important for forming the backbone of DNA. So you can just see here, again, there's a lot of steps, and so I didn't want to go through them all individually. But this just shows too how, in addition to proceeding through the glycolytic pathway to form approximately 30 ATP, that we can actually form the nucleotides, which the nucleotides are important for DNA and RNA synthesis, and they consist of a nitrogenous base. So in the case of uracil, cytosine, thymine, those would be the pyrimidine bases, or adenine or guanine, which would be the purine bases. So the nucleotides are actually a nitrogenous base that is attached to a ribose sugar, and then the phosphate group. So you can see, if we zoom in here, that these, for example, ATP is adenosine triphosphate. That's actually formed from when phosphate groups are added to AMP and ADP, which is adenosine monophosphate and adenosine diphosphate. So again, this is just the full poster, and that was just briefly about how to form the nucleotides, starting from glucose and also using CO2 to form those bases. Thanks everyone, and it was a great semester, and I hope you all enjoy the rest of your summer.